Have you ever stopped to actually look at your fingertips? I mean, really, closely look at them. Every single person on the planet, out of the nearly 8 billion of us, has a completely unique set of loops, whorls, and arches permanently etched into their hands. It's essentially the ultimate unhackable biological barcode. But how exactly does this happen? What kind of biological chaos and mathematical precision designs an identity marker so perfect that it stays with you, unchanged from the womb to the grave? Well, welcome to The Explainer. Today, we're going to unpack the biological origins, the wild history, and the genuinely mind-bending mathematics behind your fingerprints.
Okay, let's dive right into this by shattering one of the most common misconceptions out there. Do identical twins have identical fingerprints? I mean, it seems perfectly logical, right? They look exactly the same, they share DNA, they develop together in the exact same womb, but the definitive answer is no, absolutely not. Even though identical twins can perfectly mirror each other when it comes to hair color, eye color, and height, their fingerprints are completely different.
To understand why, we have to look at the massive contrast between our genetics and our environment. While twins share an incredibly close genetic makeup, in fact, a 2021 study of 381 identical twins found that 38 sets had perfectly 100% identical genes. Their fingerprints still remained entirely unique. And that's because fingerprint formation isn't just about your DNA. It's deeply dependent on microscopic, highly specific environmental factors inside the womb. Your genes, they just provide the baseline blueprint. But the environment, that's what paints the final, unpredictable picture.
Now, to really grasp this fingerprint paradox, we're going to look at a rule-breaking anomaly.
Sometimes, the absolute best way to understand a biological rule is to look at the people who completely break it. And this brings us to a fascinating, extremely rare genetic disorder called automatic leukemia. It's actually colloquially known as immigration delay disease. Just imagine having perfectly smooth finger pads, zero ridges whatsoever. Back in 2007, a Swiss woman was actually barred from entering the United States because border authorities were just completely baffled. She simply wasn't born with fingerprints. When dermatologists looked into it, they discovered that multiple members of our extended family were also entirely printless, and they eventually tracked down the single gene mutation that caused the whole shebang.
They found that these printless yet totally healthy individuals had a mutation in a gene called SMARCAD1. As you can see, this mutation interferes with RNA splicing, which essentially blocks a crucial protein from forming. And by studying this rare genetic defect, researchers basically got a lucky break. It gave them an unexpected window into normal biology, showing them exactly how genetics play that baseline role in helping a fetus's skin cells fold over one another to produce ridges in the very first place.
So let's rewind a bit. Let's go all the way back to the womb to see how this biological canvas is actually formed. If genetics are just the baseline, what actually causes all that complex folding? Well, if we follow this gestational timeline, we see that around the 7th week of fetal development, we start to grow these temporary, ephemeral structures called volar pads right on our developing fingers. Then, between the 10th and 16th weeks, that is when the magic really happens. As the fetus floats around in the amniotic sac, the soft developing skin bends and wrinkles. But here is the wild part. The exact spacing and pattern of those wrinkles are dictated by tiny microvariables. Things like the length of the umbilical cord, the overall blood flow, blood pressure, the fetus's exact position, and even the dynamics of the amniotic fluid. All of these push and pull on the skin.
Now, looking closely at this hand-drawn illustration from a forensic dental sciences review, you can see the apical and interdigital volar pads. Think of these raised, round hillocks as the foundational canvas. As the basal layer of the skin's dermis rapidly grows, it compresses and literally buckles against the softer layers above it. This buckling process, combined with the pressure of the amniotic fluid we just talked about, permanently locks the primary ridges into place. Because no two babies, not even identical twins, will ever experience the exact same womb pressures and movements at the exact same microsecond, the resulting biological canvas is literally 100% unique.
Okay. So we have this chaotic canvas. How do we make sense of it? Let's look at the typology of loops, whorls, and arches.
In the late 19th century, Sir Francis Galton and Edward Henry realized that despite infinite variations, fingerprints generally fall into 3 overarching patterns. Loops are by far the most common, making up a massive 60% of fingerprints. Whorls, those complex circular whirlpool patterns, they make up about 35%. And then you have arches, which just flow wave-like from one side to the other without turning back. Those are incredibly rare, sitting at just 5%.
But here's the thing. The overarching pattern isn't actually what identifies you. It's the micro details.
Take a look at these tiny chaotic defects here. They're called minutiae or Galton points. Notice the ridge endings, the bifurcating forks, and those tiny little dots called islands and enclosures. When a forensic analyst is matching a print, they aren't just looking at a big loop. They are mapping out the exact spatial relationship between all these tiny structural anomalies. These minutiae are what make your print a literal master key to your specific identity.
So, when did humanity actually figure this out? Let's trace the history of identification.
Humanity eventually caught on to the power of these biological canvases, and the timeline of discovery is surprisingly fast. In 1892, down in Argentina, Juan Vucetich made absolute history when he used a bloody fingerprint left on a doorframe to identify a murderer. It was the very first crime ever solved this way. Word spread like wildfire. By 1903, the New York Police Department started adopting fingerprinting, throwing out those older, way less reliable physical measurement systems. By 1924, the FBI started cataloging them on a massive scale. And then, jumping to 1999, they transitioned to the automated fingerprint identification system, digitizing millions of prints so they could be searched in a fraction of a second.
All right, now for the grand finale, the math behind the prints, or what I like to call the underlying matrix.
This is where it gets genuinely mind-bending. We've talked about the genetics and the physical pressures in the womb, but researchers have recently discovered that we can use high-level mathematics to precisely model this exact biological chaos. Look at how perfectly this illustrates the intersection. On one side, you have a real human fingerprint featuring the classic delta structure, that triangular intersection of ridges, and on the other side, mathematical classifications of those minutiae. Researchers have found that fingerprints aren't just random drawings. They're actually the result of partial differential equations playing out right on the surface of your skin.
Specifically, scientists apply what's known as the Schnackenberg reaction diffusion model. Now, this is a concept originally built on the work of the famous mathematician Alan Turing. In this model, you basically have two chemical species interacting. You've got an activator chemical that signals the skin to grow a ridge, and a substrate chemical that signals it to leave a furrow. As these chemicals diffuse across the curving skin of a fetal finger, they react with each other. A phenomenon called Turing instability kicks in, causing the chemicals to spontaneously self-organize into these incredibly complex, labyrinthine patterns.
To account for the natural curve of the fingertip, scientists use an anisotropic diffusion matrix. Now, I know that sounds incredibly dense, but what it essentially means is that the math mathematically proves the ridges will naturally grow along the specific curved directions of your finger. And when those chemical waves crash into each other, they don't just blend together smoothly, they break. This mathematical matrix proves that minutiae, those tiny islands, forks, and hooks we looked at earlier, they are not random accidents at all. They are the structurally inevitable result of mathematical chemical reactions happening in a confined curving space.
So from the genetic mutations of printless families, to the physical pressures of amniotic fluid, all the way to the mathematical inevitability of turning instabilities, your fingerprints are this stunning collision of biology, physics, and math. So I'll leave you with this final thought. Look down at your hands right now. What exact, unrepeatable combination of biological chaos and chemical mathematics perfectly designed the unique identity that you carry on your fingertips? It's a matrix that belongs only to you. Thanks for joining me on this explainer, and keep questioning the incredible world right at your fingertips.